Good morning, everyone, and thank you for joining us on our third BluVite webinar for 2021. And actually, the fourth based from data that was collected in the 2020 growing season. If you missed our previous webinars on BluVite and are interested in seeing more trial data, you can visit our YouTube channel or the Enartis USA website, where all the recordings are posted. And actually, the slide decks can be downloaded from the Enartis USA website as well. My name is JT Jaeger, and I'm the Sustainable Viticulture Specialist with Enartis USA, and I run all of the BluVite trials here in North America. And I'm actually based out of California, so most of our trial data is from the North Coast, where I live. If you've never been to one of our BluVite webinars, the format starts with a product introduction, followed by generally a deeper dive into the trial data, including canopy parameters, certain canopy measurements, and then the fruit quality parameters, which I think are more interesting. We will not answer questions during the 30-minute presentation, but we'll have a designated Q&A at the end where I'll answer any questions people may have. Feel free to add your questions to the Q&A tab as they come up, and we'll make sure to address them at the end. Before we begin, I wanted to talk about the companies that work together to make the BluVite technology work in North America and abroad. BluAgri, which is the top company there, is our Italian sister company that creates the vineyard product line, again, based out of northeastern Italy. I work for Enartis USA, and we are distributors of the product in North America. Again, we're based out of the North Coast in Windsor, California. SOP actually creates the technology that goes inside the BluVite product line that allows the product to work. And this company has been around for over 20 years and actually has many other products in staple crops like corn and soy, and actually perennial crops like almonds and sort of systems that are analogous to vineyards like olive orchards. They have peer-reviewed publications in livestock production with UC Davis, Cornell, and the University of Milan, and actually more recently had a publication released in corn production. I'm not going to talk about these today, but we're actually working pretty closely with them to achieve peer-reviewed publications in viticulture with both BluAgri and also with Enartis collected trial data. The venture into vineyards actually occurred about 6 or 7 years ago in Italy. So very briefly here, this slide shows just the sort of global presence of BluVite, all the different continents the product is being commercially applied to and also commercially trialed in. I'm based out of California, North America, United States, Mexico. The product was created in Italy, and so there's a presence in Europe, France, and Germany as well, but also more recently South America and also South Africa. Because we have this global presence, we have the opportunity to commercially trial BluVite on a lot of different varietals. The ones that are actually in bold are varietals that I've worked directly with here in North America and California, Oregon. Specifically, today we're going to talk about a Cabernet Sauvignon trial. The presentation did say 2nd and 3rd year data, but for simplicity's sake, I'm going to focus in on the 3rd year trial data, the more recent data that was collected. So one of the problems really of being a plant or grapevine is you can't move. So plants really have to use soil microorganisms to make a living, to exist, to thrive in the surrounding soils. Plants are dependent on the growth of these soil microorganisms, which ultimately possess the sort of metabolic machinery to access soil nutrients, especially the ones that are minimally plant bioavailable. Nitrogen and phosphorus come to mind. There are also some very complex interactions that take place in the soil between plant roots and these root-associated microorganisms in the rhizosphere, which is what it's called, the area that root microbial interaction occurs in the rhizosphere. The soil microbial loop is the reinforcing cycle that results in the emission of fine lateral roots, and I'll show some slides on our nursery trials. But really these secondary roots that are responsible for the majority of the nutrient and water uptake for grapevines. These secondary roots are hugely important for the longevity of the vine, especially through extreme weather events like drought, for example. And we'll dive into some of this data that was collected from the third-year trial that we're talking about today. And because, really the vine can then better utilize its water and nutrient resources and actually be more resilient through these events that we're speaking about. And actually these events becoming more and more common too as well, especially on the west coast of the United States. Microorganisms are really a critical piece of this loop, or essential. They're an essential piece of this loop, which take these nutrients that are plant unavailable and through different processes like natural population turnover become available to the plant. And so the BluVite product line is actually directly stimulating this root microbial interaction within the rhizosphere. Moving on to our nursery trials to sort of look at secondary reproduction and how that relates to our mature vineyard trials. We've done extensive grapevine nursery trials in California and in Italy as well, collected by both the Enartis team, but also BluAgri and Martina Brogio, who's my colleague in Italy with BluAgri. And you can see from here, this is just the vegetative growth of these grapevines, these young Merlot vines. But we also did extensive measurements of the overall rooting biomass, the subsequent rooting biomass of these different vines treated with the BluVite product. So now we're actually looking at the overall rooting biomass of the two treatments. This is the same nursery trial that we covered in the previous slide. But now you can see the increase in the overall rooting biomass. Not only that, but also the increase in terms of the amount of secondary roots that are occurring in this nursery trial with the treatment. It's pretty marked effects on the quantity of these secondary roots, which are hugely important for nutrient and water uptake. This is just a different angle of the same, different vines from the same treatment and the same trial. And this kind of gives another indication of what I'm talking about in terms of the amount of secondary roots that are produced and the overall rooting biomass that is actually greater in the BluVite treatment relative to the control. So for the presentation today, I pulled data from a third-year Lake County trial from 2020 growing season that I'm going to actually dive in a bit more depth today. And this slide is really all the data that was collected in 2020 as part of this trial. And I really won't be able to go over all this data. It's too much data to present in one session, especially with a 30-minute time limit. But I did pull out some interesting interesting sample points from a handful of these time points that I think will sort of get the point across and tell a story about what's actually occurring in our trials. So this data was actually collected during the bud break, the period of early shoot development and moving into rapid shoot elongation later on in the season. But this is really a critical time for the initial, programming of vine canopy architecture and then actually the resulting future water needs of the grapevine. So this is kind of an important time frame to cover as part of this presentation. But before we jump into the actual trial data itself, just a little bit background as to the application of the product and when we do that. So BluVite is applied twice a year, once at bud break and then once at pre-bloom. And generally in the Northern Hemisphere, that's March, April for that first application, May, June for that second application. And really, we target the application at this time, because it's the most effective time to stimulate microbial root interactions when uptake of soil nutrients and accumulation of these nutrients are very active, particularly active. And this is really so we can influence vine development for the rest of the season. There's also this multi-year influence, so multi-year application, that in basically the treated vines are better able to store nutrients from the previous year, which is one of the main drivers of this uniform bud break that we see in the treated areas relative to the control. Now moving on to canopy observations and measurements. And generally in vineyard systems, variability of shoot growth is common. And the red bars indicate sort of the length of the shoots across the cordon. You can see here this variability I'm talking about. These red bars do indicate a high level of variability of shoots at bud break in this particular block in the control. And as you can see here, there is more uniformity of growth of shoots across the vine in the treated sections of the vineyard, with this less variation between, I guess, growth of any shoot on any given spur position. And really, with multiple years of application, we see that this variability of early season shoot growth is far less characteristic of the treated area of the vineyard in relation to the control. So now actually getting into the trial data itself. So these are all the shoot length measurements from this particular trial block. From the BluVite red treated is on the left-hand side. The control is on the right-hand side. And this essentially is all the BudBreak measurements from the vines that you saw in the images in the previous slide. And in the BluVite treated area, there's generally a 14.8% increase in average shoot length. But this is really in conjunction with the decrease in shoot length variability in the treated area when compared to the control, a significant decrease. So really this increased root biomass and the secondary root production early on in the season allows for this higher root shoot growth functional equilibrium later in the season. And we ultimately see improved ability of the vine to utilize soil nutrients and water resource with more uniformly developed canopy. And this all starts from the early season programming that we see here. This overall increase decrease rather, of short thin shoots in the treated area relative to the control. Now, moving on to the May measurements around bloom, post-bloom. And this really is when the vine reaches its full canopy size. This vegetative growth begins to slow down, and really the vine transitions from vegetative growth really into reproductive growth. And I'll talk about a few different aspects of the measurements that we took during this time. Again, I used another side-by-side comparison of images here just to give you an idea of the different canopy sizes, average canopy sizes in the treated versus the control. You can see here that we actually have a higher functional equilibrium between sort of root growth and shoot growth in the treated. You know, this also includes mechanisms that control the distribution of photosynthetic compounds or photosynthates to the different sinks in the grapevine, which include the shoots, the roots, and the fruit. Overall vine balance in the treated area has implications for the vine's capacity to fix carbohydrates. These are actually created during photosynthesis, and then accumulate nutrients for growth for later on in the season. Ultimately, you know, these fixed carbohydrates will be super critical for the future ripening of that fruit. And actually, because of this, it has implications for fruit quality and yields. Again, this is the same format of the data that I showed in the previous slide. Hundreds and hundreds of data points per treatment that were collected, and this was shown on the same graph here. And this really equates to about a 23.2% increase in average shoot length in the treated versus control. Again, the treated is on the left-hand side, the control is on the right-hand side. But you can see it's not due to excess vigor. There's not a huge difference between the full extent of the shoots, but you see there's far less shorter shoots in the treated versus control. And this is due to a general decrease in shoot length variability in the treated vines compared to the control. There's a much smaller standard deviation. It's actually about 20 centimeters in the control versus about 14 centimeters in the treated area. And we see a pretty consistent decrease in the shoot length variability it begins with BudBreak. We see it at BudBreak, and then that shifts the overall canopy size upward later on in the season. And this really gives us a clear indication of more uniform growth. And then this equates to more efficient vine transpiration in stomatal conductance within a vine and actually between vines. And this really has huge implications for late season water stress levels and also for thermoregulation of the canopy, which I will cover in this presentation at this particular trial site. Now moving on to leaf nitrogen levels, and I guess more directly, the measurement of photosynthetic compounds using chlorophyll meters. So a chlorophyll meter really is used to guide nitrogen management in many cropping systems, and it does this by monitoring leaf nitrogen status. Researchers have found that actually SPAD values are significantly correlated to leaf nitrogen levels in vineyards and also leaf phosphorus concentrations. And so we generally measure plant nutrient status or vine nutrient status at bloom and veraison because approximately 40% of nitrogen use in the grapevine occurs from fruit set to berry softening. So it's a critically important nutrient at this particular growing stage. Similar to what we see in the physical canopy measurements is this more uniform average leaf chlorophyll content in the BluVite red area relative to control, ultimately with a higher average chlorophyll content in the treated. And this can be directly correlated to higher leaf nitrogen content at a stage really when nitrogen use is at its highest in the grapevine, essentially fruit set through lag phase. Moving on to the August sample points, really at post-veraison is the grapevine phenological stage, but really this is a critical irrigation period for the grapevine. Once you're through veraison, you don't really want to have any water stress being applied to the grapevines. And really this ultimately will determine whether the vines are better able to handle the water needs. Infrared thermography has really been proven to be a rapid and reliable indicator of canopy temperatures and can be an indicator of water usage in the vine and actually water use efficiency through heat waves. So this is data collected using a FLIR thermal imaging camera, and you can see it's broken up by trial set, and then there's an overall average as well on the far right hand side of the graph. This is given in temperature differences between the treated and the control. So if you have a negative difference, it's a decrease in canopy temperatures in the treated. If there was a positive, it would be increase. So we actually see overall decrease of about 0.67 degrees Fahrenheit in the treated, measured in the treated when compared to the control. And that's across all trial sets. You can see in set 3, it's actually a more drastic decrease of 1 degree Fahrenheit. Now we want to look at rates of stomatal conductance. And stomatal conductance is related to the opening and closing of the stomata on the leaves of the grapevine, which is directly related to plant transpiration. In the data on this chart, we see nearly 35% overall average increase in the rates of stomatal conductance in the treated when compared to the control. And really because the BluVite has an overall average larger canopy size with higher rates of transpiration at this critical phase, this does allow for a higher cooling effect in the canopy overall. And really this ultimately means that the vines are better able to regulate their temperatures through these high heat wave events like we experienced in 2020. Now we're really going to dive into certain fruit quality parameters. And today I picked out some important ones like potassium and pH and how they relate to each other, but then also looking at yield data through these late season heat, but mostly this like late season water stress that we tend to see in some of our vineyard trials, especially as this vineyard is planted on 101-14 rootstock. Yeah, and generally 101-14 rootstock is not really that drought tolerant, so that's why this trial is an interesting one. Here are all the analyses that we measured over the season for this trial. And again, like I said, I've chosen some more interesting data points for the purpose of today. I physically cannot go over, again, all the data points that were collected as part of this trial. There's just too much data to cover in one little half an hour session. So yeah, again, I've chosen the ones in bold to pull out. First, we'll cover some yield analyses, and then and how that relates to dehydration, and then we'll go into potassium and pH. So now looking at two comparative examples, I guess two pictures from the treated section and the control. And I'll show the data for this. So, obviously these pictures are just pictures. Data can be more impactful, but I think also showing images can be impactful as to what's happening. I'm going to also show in the next slide sugar per berry data. And these calculations are really a function of berry volume and sugar concentration. So if you just show BRIX, I don't think you're really getting the full picture, because once phloem transport is ended, any further increase in BRIX is due to water loss or dehydration and not continued synthesis, which is what the sugar per berry data can show you. So, berry weights really can be monitored to assess the onset of dehydration. And this is when average berry weights decrease while BRIX increases, but then sugar per berry data can give you further validation of that assessment. So here are the berry weights from this trial, 2 sample points, both in September up until harvest, which was in mid-September. And you have the same format. Control is in gray, BluVite red is in a pale red color. And you can see that berry weights significantly decrease from September 3rd to September 15th for the control vineyard, but berry weights are more or less maintained in the treated. And this results in overall higher berry weight averages prior to harvest, which again was in mid-September. Now moving on to the BRIX data, the following slide, the next slide I will show is going to be sugar per berry by volume. But if you look at the BRIX data, you can see that in both treatments there's a significant increase in BRIX over time, but a more rapid increase in the control versus the treated. Ultimately, prior to harvest, BRIX maxes out at about 30 in the control and just over 28 in the treated. And this is really where the sugar per berry by volume is critical in figuring out what's actually occurring. Is this due to dehydration, or is this due to actual synthesis of sugar? Now looking at the sugar per berry by volume data, you can actually see from the 2 different sample points that there is no statistical difference in sugar per berry by volume over time in the control, indicating that increases in BRIX that we saw on the previous slide are not due to active sugar loading, but dehydration. And we also see that the synthesis of sugar continues to occur just up until harvest in the treated. So what we see here is a significant decrease in berry weights and a significant increase in BRIX with a lower sugar per berry by volume in the control. And this really indicates that dehydration was more apparent in the control area of the vineyard. Also, we see a higher sugar per berry volume concentration in the treated prior to harvest, which implies that overall synthesis of sugar was higher in the treatment, resulting in those increases in BRIX. So now moving on to the vine water status and how that it relates to potassium accumulation and then resulting fruit pH in particular. And this is actually late season vine water stress. Based on the crop water stress index calculation, you can see there is an average of about 26.7% decrease in the values in the treated area of the vineyard relative to the control. And ultimately, the treated vines will use more water during peak leaf area because of this increase in uniformity, which ultimately shifts the canopy size to become sort of overall larger, which results in higher rates of transpiration and photosynthesis. And so with that in mind, we don't see an increase in irrigation needs, and oftentimes the irrigation regimes in the treated and the control are kept the same. And in this particular case, they were kept the same. So really this overall root biomass allows for the vine to explore a larger soil volume and utilize its existing water resource more efficiently. And because of this, the vine experiences less late season water stress through these drought conditions, and then 2021 is going to be another year in which we monitor this really closely. But the point of mentioning this is this has huge implications for fruit quality parameters like potassium and how this is related to resulting fruit pH. Now looking at the potassium data from the same 2 sample points, we can see potassium accumulation occur in the control relative to the treated. And potassium channels, especially during late season water stress, tend to be upregulated, and this plays a major role in potassium loading into berry tissues. Grapevines will accumulate potassium in clusters to decrease the cluster's sensitivity to drought. Potassium can maintain tissue turgidity or tissue turgor and remedy any oxidative stresses that might occur during these events. So an increase in drought or water stress can mean a greater increase in cluster potassium levels. And so from this data, we see that we do have a significant decrease in potassium levels in the treated versus the control, which does have implications for less late season water stress related potassium accumulation in our treated area of the vineyard. So moving on from that, during the ripening process, fruit pH will generally increase as you approach harvest ripeness. And so you can see here that in both the treated and the control, there's a general increase over time. There's a handful of reasons why you would lose acidity during fruit maturation, including lower rates of acid synthesis or loss of acid due to various mechanisms that I won't get into today. It's a whole presentation in itself. More interestingly, pH can increase due to this influx of potassium that we saw in the control on the previous slide, especially in the period of late season ripening. So tying in the previous slide, we saw roughly 22% decrease in potassium concentrations in the treated block just before harvest, which can largely explain these pH differences in our trial areas. So that concludes the data presentation portion of this presentation. And so now I'm just going to cover the trial summary, and then we'll move on to the question and answer portion. So increased root biomass and secondary root production allows for this higher root shoot growth functional equilibrium that I talked about. And we see this in the root biomass in our nursery trials, and we see this, in the overall vegetative growth, the canopy growth in our vineyard trials. This goes hand in hand with an increase in vine transpiration and stomatal conductance, which allows for better cooling potential during high heat wave events. And this is represented by the thermal images and the stomatal conductance analysis. With this in mind, there is a potential for the larger, quote unquote, larger canopy because of this increase in the uniformity of growth for higher rates of transpiration. And so, vine may use more water during the season. But we actually see in oftentimes cases decrease in late season water stress. So this is supported by this increased root biomass and the improved water use efficiency. So the grapevines ultimately can use this existing water resource more efficiently. We also see a decrease in late season berry dehydration and in potassium accumulation as well. And this goes hand in hand with these late season water stress conditions and high heat events that we experienced this year in 2020, or I guess last year in 2020. So now begins the question and answer portion of this webinar, and I will go into the question and answer box now and answer the questions that were asked in the order they were asked in. And so if you haven't put your questions in there yet, just go ahead and add those to the question and answer box tab that you can see on the right hand side of your screen. And I guess we'll begin that now.